Thank you, students, for that. Does it echo? And it's, in fact, my great honor to welcome and introduce Professor Kasturi Venkateshwaran to all of you. So, I and Dr. Kasturi happen to collaborate or connect in a rare incident. That was a case where we described one bacteria from the urine of a patient, and while he has described a few months ago from the space. So that is from the space to the human connection made, and then that is where we just collaborated and it has been taken. And right now he is here with us to share his experience. So for those students, graduate students, as well as my colleagues, it is my pleasure to introduce Professor Kasturi Venkateshwaran. So he is currently senior research scientist in Caltech in the Jet Propulsion Laboratory of NASA, and he supports the biotechnology and planetary protection group. He has 44 plus years of experience that encompasses marine, food, environmental, microbiology, and many others among them. He is also having ISS microbial observatory projects to measure the microorganisms associated with the US nodes as well as the Kibo Japanese experimental modules. And as grad students are there, like how a journey began for him, like he's like all of us studying in India from Indian universities, and like how his journey has gone into the prestigious NASA. I'll just read about his education and experience for you all. Yeah. He did in 1977 MS in marine biology from Annamalai University, and then in 1982 PhD in marine microbiology from Annamalai University, and subsequently he did another PhD in doctorate in agriculture in Hiroshima University, Japan. And he started his career as a junior scientific assistant export inspection agency of Ministry of Commerce, India, between 1981 and 86. And subsequently, after 1990 to 1993, he became senior scientist Marine Biotechnology Institute, that is the JAMSTEC, Japan. And 1993 to 1996, he also served as chief scientist Nippon Suizan Kaiser, Japan. And afterwards, in 1996 to 97, he served as senior researcher in University of Wisconsin, Milwaukee. He systematically arranged phylogenetic affiliation of Shivanella that time. That's a bacteria you may be knowing. And subsequently, he became in 1998 to 1999 senior scientist astrobiology in Caltech. And currently, from 1999, he is senior research scientist just in the JPL lab of Caltech, NASA. And towards his contribution, I'm sure we will be going to listen it from him rather than me making kind of some kind of readout from the available stuff. But it's better like you all will understand the depth of knowledge that he has and the contribution he has done to the field. So briefly, he has applied his research in molecular microbial analysis to better understand the ecological aspects of microbes while conducting field studies in several extreme environments. He started his work in Japan, that is in JAMSTEC, on to the deep sea explorations, and then spacecraft mission, that is of the Mars Odyssey, Genesis, Mars Exploration Rovers, Mars Express, and many others, and assembly facility clean rooms for various NASA and European Space Agency facilities and the space environment on Earth in Earth orbit. And he also directs several research and developmental tasks for JPL Mars Program Office, which enables the cleaning, sterilization, validation of spacecraft components. He directs several NASA competitive awards on microbial monitoring, spacecraft, and associated environments for the Exploration System Mission Directorate, closed habitat like ISS or its Earth analogs for human exploration, and Operation Mission Directorate. He also provides expertise for non-NASA programs such as commercial agencies, medical industries in measuring microbial pathogens that are problematic to health-related issues. And our conversation with Dr. Kasturi Venkateshwaran from yesterday to today has explored many other fields, what he is like. He is also having a lot of technology developed, including for DNA sequencing from samples and many other newer technologies, which he is going to share with all of us, his journey and his work that is dealing with the various aspects of microbial, microbiology, microbiome, as well as the pathogenicity, virulence, as well as technologies for monitoring monitoring the microbial load in various like the space or various systems. So it's great honor for us to have you here, and it's pleasure. I'm sure all of you will enjoy listening to him, as well as you may have many messages to take home. So I take this opportunity also to welcome Dean Medical College, Vice Dean Medical College, my colleagues from other colleges, undergrad students, the BSc Microbiology students, the PhD scholars from our center, as well as from other departments, and faculty and technology incubator people where we will be visiting in the afternoon to have more discussions, and other innovators and my colleagues. Please, sir, thank you.
Thank you, Dr. Rekha, for sharing the brief introduction and his expertise with us. We now request Dr. Abhay Nirgude, Professor of Public Health and Dean of Yenepoya Medical College, Mangalore, to honor the chief guest, Dr. Kasturi Venkateshwara.
Thank you.
Thank you, sir. Moving on to the most awaited moment of the day, as we all are eager to listen to Dr. Kasturi Venkateshwara on the topic Microbes in Space, Challenges and Solution for Omics in Space. Over to you, sir.
You have to switch to this, right? See that? Swap. You have to swap. Yeah. Let me see. No, there. No, see that? You make it bigger? Okay. Thank you very much, Dr. Rekha, to invite me here. It's a wonderful place that I'm enjoying already with the interaction with the students, and you have a phenomenal facility, and just thankful for the invitation, you and Dr. Arun. So I will also tell about my collaboration with Dr. Rekha and her group, how it all started, later in my slide. So this slide always I open up to show you what we are doing in NASA Jet Propulsion Lab. So as you see here, this is a small Pathfinder in 1997 that landed in Mars. I don't know how many of you know this. This is a very small tie, about less than a kilogram, weight. And during that time, we had a different system to land in Mars. So the biggest issue for exploration of other planet is how you land in other planet. You can cruise to the level as much as you want. The engineering system and the technology were matured enough for cruising anywhere you want, but precisely landing in a specific planet is a big issue. It is called EDL, entry, descent, and landing. It is something we call 7 minutes terror. Most of the women may know, it is just like I give birth to a child, it is so much of complications there when you precisely land in place. So 7 minutes of terror is there. Anyway, so that 1 kilogram level to 1 ton size, right now what is roving in Mars is Mars 2020. What you are seeing here is Mars Science Lab rover. It is not lander, it is rover. Okay? This kind of the system that you are going and landing in other planet require so many engineering things, that is fine, but if you are going for detection of life in other planet, better you clean those things. Otherwise, what you end up with is whatever you are carrying with you is also life. So you will be compromising the science. So these are not cheap. This $2.5 billion worth of engineering effort, each mission, this kind of mission cost. So the one thing since 1997 to today is what is not changed is not the size, but the requirement. For example, how you can clean a system in those days, 1997, is the exact same requirements no matter what the size is. It is, think about it, 1 liter of water contain about 100 microbes. Think about that. That is your requirement. Okay? But even if you take 1,000 liter of something, should contain only 100 microbes. So that means whatever the size it is, you have to reduce your microbial burden to that level. Otherwise, you are dumping your materials in the exploring planet. Think about, you are going for a national forest. They tell you, carry along with you whatever you are taking with you, bring it back. Don't leave anything on the place that you are visiting. Same thing here. We should not take anything unwanted material. We should not contaminate other planet. So if you contaminate the planet, then you are compromising the science. So today I'm going to tell you something about omics in space, say microbes in space. What are the challenges and solutions that we are facing to do that omics in space? Most of you know what is omics, but I'm just going to tell you, omics is study of molecules. Either it is DNA, genomics, protein proteomics, lipidomics. Although omics is the study of the molecules, because we are in a DNA era. If you are exploring things in space, better you know this kind of technology. Okay, so that is my contact number and other email addresses. If you want, you can take a note of it. You can get back to me whenever you want it. And I'm not going to touch bases because of the time. I'm not going to tell what I have done deep sea to deep space, anything in between. But I will touch only what I have done on this International Space Station basically, or some of the Mars rover that we are working on. So some thoughts, people will think, hey, you are in an engineering institution, why the hell you are doing biology? People will think about that, right? Because, as she started saying, we have to dream big. Why do we need to do this exploration? We need to dream big. Are we alone? Where did we come from? Still, some of the people who are doing the phylogeny of the life, the tree of life, still we do not know the root where we are from. Obviously, we came from microbes during the evolution. Things like 4.2 billion years back, the microbes formed. It might have formed with the fusion of the physics and chemistry. Physical nature combined with the chemistry might have formed biology like RNA, etc. Or we get seeded from other planet. We might have come from the planet like Mars via meteorite, not as a human, but as microbes in those days. So these two theories going hands in hand. The one that gets seeded called panspermia. panspermia. Okay, the one that is evolved with the physical condition and chemical condition are different story. So how RNA formed from there, how the life formed, etc., etc. So microbiology. Why we are doing microbiology in NASA? We are asking these questions. 99% of our work in NASA is engineering. That means you are asking the big question, and that has to come true. There is a process. That process is almost 99% of it is engineering feat. So you need engineering system and engineering stuff to make your dream come true. So one of them is you go and look for life on another planet using instruments. If you are going to look for life with a biomolecule, better you don't take those biomolecules along with your probe. Clean them to that level so that you won't get mixed, unless you are looking for non-earth centric life. What that mean? We only think life based on carbon. We are not thinking life based on silica or something. Our imagination is thinking that the life that we know is based on carbon, protein, lipids, sugar. So these are the backbone of life. So if you are thinking like that, so the probes that you are making will also contain those kind of things that you know what it is before you send. The hitchhikers, that's what is called planetary protection. You take a passenger list of the spacecraft that you are sending out to measure any kind of microbial life in other planet. Or tomorrow if you are sending human to moon and Mars, moon is fine because we already did so many work and we came to know there was no life. But the one we are targeting Mars and the moons of the Jupiters and other things, there may be likelihood of life is still there. Okay, so tomorrow when you are taking the human in there, you will lose all this contamination issue because you and me are the walking fermenters. We carry more microbes than our microbial cell, our human cells. At least 100 times more microbial cells there than your own cell. That means you have microbes. Don't think that most of you know, some of you are actually microbiologists. Microbes are not considered as bad. It is required. But if you tell the normal people microbe means, they always think it is bad. But it is not. You need microbes to digest your own food that you are eating. Whatever you are breathing and inhaling and breathing out are containing some microbes. We all know, experience the corona, that 6 feet away and all this kind of stuff. You're touching things, and also we know how to leave behind certain things so that it is actually contaminating other places. So what I'm trying to say is, the good microbe that you have, you need to find out whether that is useful for human exploration. For human exploration, you cannot simply take the water for whatever things you want. You have to make water with the resources available there. With the use of microbes, it can be done. You have to extract materials out of some, trash, for example. Even now we are taking the atmospheric condensation from there, we are extracting water out and use it in physical means. We are actually extracting water out of the urine in space, and then that is recycled. So all these things can happen with the help of microbes. So you need to know how the microbes behave under space conditions. The most important things are against microbes are microgravity and enhanced radiation. But microbes are clever. They were there for 4.2 billion years back, right? They are very clever how to adapt the conditions. They know what the unfavorable conditions are and shed most of their metabolism, but kick something else to defend themselves. That's why antibiotic resistance increased. That's why virulence increased. Okay? So the other topic that I'm going to touch today is novel microbes. This is what I was also working with Dr. Rekha and her group, and I will also tell about that today. And characteristics of novel and unusual microbes, something like biofilm, and how whole genome sequences are being used, and then proteomes, metabolome, and etc. And one important thing that you all need to know, that even on the earth, that we don't know more than 1% of microbes. 99% of the microbes that we eat, we live with, we don't know. How do you come to know? That's why people in clinical area also, they say emerging pathogens. They don't know what it is. SARS emerged recently. It was not, it didn't come from other planet. It has been modified and mutated to the thing that we faced. So those kind of emerging pathogens can be, characterized using metagenome. If you come to know the characteristics of these microbes using this sophisticated technology, metagenome, you know the functionality of those microbes as well. Then now you can tune to the level and then culture them in an appropriate condition. That's how we got something and then published, and then later on Dr. Rekha also found in some other patients, and we started getting more and more from other patients. Until then it was termed as a emerging pathogen. Here I want to show you how the planetary protection is doing. So she is my boss now, but she was my student 15 years back. So young girls, you have to think about, work hard, and you can be a boss for your own mentor. Okay, I hope that you are going to hear.
It's really important that we send a rover that's clean and we make sure that it doesn't contaminate Mars. My name is Mujige Stricker and I protect Mars from Earth bacteria. The next Mars rover is slated to go to Mars, collect samples so that...
Is it coming? Okay.
We send a rover that's clean and we make sure that it doesn't contaminate Mars. My name is Mujige Stricker and I protect Mars from Earth bacteria. The next Mars rover is slated to go to Mars, collect samples so that eventually we can bring those samples back to Earth and determine for the very first time, did life exist on Mars? There's nothing that we can build that's sterile. So we take swabs and wipes of the spacecraft as it's being built. It gets put in an oven, it gets put in various chambers and clean rooms so that we can maintain that level of cleanliness. If we do find something on Mars, we have to make sure it's something that actually came from Mars and not something that hitched a ride.
So
this is the place where the magic happens.
Where we
Definitely cool.
measure
In this lab, we look specifically at spores. So spores are those hardy microorganisms that
Mars
can actually
rover
survive
surfaces
if it made it on the spacecraft and the
sent out,
journey through
we have
space,
different
through
stages.
vacuum. It's very
stages. We
humbling
go
to be
and
a part of
collect
this big
microbes and
project
see
because there
how
are
much
hundreds of
load it
people
is
that have to
because
come
we have a
together and
requirement,
build a
right?
spacecraft. There is no one person
We
that
have
can
a
say,
requirement
I did
saying
this,
that
I made
only
this happen. It's always
this
a
much
we.
microbe you
I
can
owe
carry
it all
with
to
you.
Carl Sagan and watching the Cosmos.
So at
I
every
remember being a
single
little kid,
stage,
going
we
to the
have to go
public
and collect
library and
samples
renting that
and then,
VHS. And from that
account
moment, the
the
light bulb
microbial
turned on. It
content
actually was the
before
start
sending it out to
of
the
my passion of
space.
science communication.
So it is a
We are citizens of
5-year
our
process.
universe. We have to be good ambassadors when we are
So
exploring other planets,
that's
other moons.
one thing
So it's
I
the right
did not
thing
know.
to do.
Because of the audio issue, you are not able to hear what this video is saying. But basically, this is the planetary protection content of what we do. The other one is Mars 2020 rover.
It's really
how
important.
it drill and bring the sample back. If anybody tells you that we already got the samples from Mars, that is a lie. We have no samples from Mars. We got samples from Moon. That's not a problem, and that's why we ruled out there is any life in in Moon. But there is likelihood of life in Mars, then we have two issues. One is how to keep the Mars as is, as pristine as it is. The other one is whether you bring the sample that contains some Martian that actually make us get extinct. That's called back contamination. You have to have extra care until you know there is nothing. But science is what? It can only definitely say there is something in it, but we can never say there is nothing in it. It is undetected with the technology that we know. Think about, my days in 1981, I did not have a fax that transmit the photograph. When you apply the position and all, we did not have. But now instantly you are sending it out. Even 10 years back, Google was in the infant stage, they cannot connect with their loved ones. But now, as a girlfriend or boyfriend, if they are not calling you in the night before you are going to bed, you will be angry because he or she cannot deny that they don't have a technology. They cannot have contact information, right? So the technology is developed that much. Now tomorrow it will be different, right? So that is the thing here. Now we don't have a sample yet because we don't have a technology how to tackle the back contamination. But we don't have a technology how to bring samples from Mars. We know how to land. We do not know how to fly from Mars. So let us instead of wasting the time, let's go and then collect the samples and leave it in the Mars until you develop a technology to bring it back. I don't think we will have a launch pad in Mars ever, at least in my lifetime, unless human go there and build it. Human are planned to go there to have a sustained living in 2034 or something. But until then, you cannot simply keep quiet. So you collect the samples and leave it. Maybe the orbiter that orbiting Mars can go and fetch it like a helicopter, fetch it, and then come back to the orbiter, and then from there it can bring it back. That is the technology we are planning to bring the sample back in 2030. But Elon Musk was telling that he can bring it 2023. It is a stunt. It is not happening yet. Okay? So this one is that how samples are collected. Again, there may be some issue with the audio. That is fine, but you can think about the rover. I think there is no audio here. This is the rover after landing. These are real pictures. These are animated pictures, and then this is clearly showing how it is going, and then collecting samples. Think about all the geometries. Every single thing we have to account the microbial presence. Every geometry, every electronics, every optics, every cards, electrical cards, and all these things. It is going to look for appropriate place, and then sensor is telling, yeah, there may be something here, because they have some spectroscopy that tells, okay, this may be interesting thing, and then they drill it, and then the drill is going to get into this suitable tubes, but I don't know why it is not going. I'm seeing here it's not there. It's there is some problem. But this is the tube that hermetically sealed, that went through 500 degrees centigrade. That means you have a material. Some of the material scientists here may understand what I'm talking about. You cannot cook everything. If you cook, the electronics will not work. Like operation successful, patient died kind of situation. We don't want that. So material compatibility of your technology is very, very important, but this particular thing is more important. That's why the tubes, sample collection tubes, are heat to 500 degrees centigrade. That means we know right now no technology that could detect anything, even biomolecules get everything pulverized. So nothing is left behind. So those are the things we are putting together and then leave there and then wait for the next technology to come to fetch it from there and come. We are expecting 2034 is the one that we will be getting it back. Okay? So these are some of the introduction for Mars thing. Let's take you to space station. Anybody knows this is the red guy? It is written there. This is the guy lived longest time in space station, Scott Kelly. His identical twin was in the ground. So we were looking for various microbiome of this twin in space versus here, as well as their physiological human genome and other things, how it is different. Identical twins, but still they are different, and also they, whatever you are eating is the one that dictates the microbiome, the gut content, gut microbiome. Scott Kelly, poor guy, can eat only the dry food because you don't have ice cream up there unless you make it in space. And then the Mark Kelly, who is his identical twin in Arizona, he is a senator actually. He can enjoy even the steak and other things on the ground. That is the only difference, but the rest of them are same, and they wanted to compare these two, what is, psychologically and physiologically and genetically, these two people are different. So around 565 days, 350 days, less than a year, 350 days, he was in space. And how things change, and they collected around more than 100 samples from him, blood samples, urine, tissues, and all the things, and then see how it is. So this picture also shows that they are eating the brownie. The brownie also comes with penicillium. So that means it is floating around in space station, which is close. It is very close to habitat, right? This is the guy, Terry Wirth, who collected my samples. For the past 9 years, I collected samples, and the very first 2 samples he collected for me. After that, various different astronauts collected for me. Okay, so this is how it looked. And this is a 9 minutes video. I'm not going to show you all 9 minutes. The very important thing is, even today, I'm impressed with Dr. Rekha's lab. When you get into this microbiology lab, you need to take appropriate precaution and have the PPE, personal protective equipment. That means you are not contaminating into the lab too much. Okay, they are also same, but they live together in a closed system. This is the first time ever, they were wearing the glove when they collect the sample. Okay, this is the one that you are seeing here. I think you are seeing my mouse. This is the air sampling device. You put it there continuously for 15 to 25 minutes and then collect around 750 liters of the air and then bring back that filter and then see what is there in the air at the various different locations. Okay, and he started collecting environmental samples, wiping the environment around there. Why it's not coming? Okay, so you also take all the photographs, everything. You don't have a photographers like a dedicated photographer, right? We have here everything they have to do. They are sophisticated janitor. They have to clean their own, things. They have to clean their system, everything. So now, after seeing that he is not wearing the glove but collecting samples, then we said, okay, wait. We talk to them directly whenever we want, and whatever things they are not doing according to the protocol, then he started wearing the glove. You and me are wearing the glove during the corona time. We find it very, very difficult, right? Even in the Earth, but in space station, everything is flying and floating. It is not an easy job, okay? But still we insist, otherwise we are going to get his skin microbiome, right? So he's taking few minutes. And one more thing, crew time is valuable. That means every 1 hour of their time, it costs minimum of $2 million. It's not astronauts get that $2 million as their paycheck. Think about you're flying, think about all the other instruments needed, all the things maintained. So all those things cost together roughly $2 million per hour of the crew time. That means you have to intelligently use them. It is not only your experiment going on. At a given single day, they have to perform minimum 50 to 60 different experiments. So their time is appropriately chopped and then organized. Okay? So that's how he collects the samples after wearing things, and around roughly 1 meter square surface area come back to the earth and analyze. Okay? And one other thing I want to show you here is one second. Okay, so when he is collecting samples, since other people are also living, here is Mark Kelly is swimming around him. That means he is shedding his microbe. That is also get collected. Okay, so in some other time, what happened? You get a chatterbox. That means some other person next to you will talk to each other, because you cannot say that person, don't come near to me, I am collecting samples. So there's some chatterbox out there to talk more, and then their oral microbiome also get covered. So this is an example I'm giving you. So today I'm going to touch bases with the biofilm formation in space and enhanced resistance. Okay, and then we also develop some antimicrobial coating that is useful for that, and then how do you mine the data that you have collected and then get some novel microbe. This is the Calamia astoria I'm going to touch. And then more importantly, you cannot always say, send the samples back, I have a best available instrumentation down on the earth, I will do it and send this results back to you. Or you don't have a FedEx system. You don't have a DHL to go and then bring the sample whenever you want. So it is always good if you explore further beyond our solar system or our Earth system. Right now we are doing the low Earth orbit, which is space station. You can reach there in 5 days and get the samples back. If anything go bad to the human, you can bring them back. But if you are thinking about Mars, it is not easy. One way is 9 months. Immediately if you want to come back, you cannot come back within 9 months because you are in a different orbit. You have to wait for another year so that the Earth is coming closer to the Mars. So totally it takes around 2 years. So you have to do everything in there. Such capability you have to develop. So I'll tell you what is in practice today and what are the challenges to overcome and what are the capabilities we have developed, something like samples to sequence strategy. As I already told you, I'm not going to cover everything that you are seeing here. We also do appropriate ground control, like in a simulation facility that tells you the mimicking the space station except the microgravity. We have a hermetically sealed system that people can live for 500 days without getting connected to the external thing. And then they have to grow their own food. They have to recycle their own system. All those things without getting connected with the outside the world. 500 days, how they behave between each other. Think about how much chaos we have gone through when we are locked down. We could not go out from our own home. So we are mentally disturbed. We could not go out and eat. We could not go and meet people, right? Similar thing will happen. So 500 days, we have simulation facilities, how it is happening, including not only the physiological thing, mental, psychologically, how they are affecting. And then I can talk about the inside. I'll talk about what is happening in the omics in space. And I'm not going to touch the drug discovery. Anybody interested, we can talk. Intentionally, you can take some of the microbes in a contained way and then do some of the things that whether microgravity influence the production of certain things. So those are intentional exposure. And for space biology to understand this space station is a big real estate that you have outside the space station. You can mount certain microbial content in a unique way, so that you can expose longer period of time and bring it back to see the study. So that is what is exposure to outside, that I'm not going to talk. Anybody interested, let me know. And then the space station built 22 years back. So that means 22 years of radiation there. So is there any microbe that hitchhiked and then there is still there, and that may be a good clue for us for the future astrobiological process. Okay, so I will talk about inside the space station and omics in space. That means how you can measure omics in the space, what are the capabilities you need. We actually came a long way from simple, growing on a plate to metagenome and various different. We used all these things at various different stages to measure quickly, rapidly, so that you help the engineers to build the spacecraft that I showed you before. That took about 5 years from the day one to the final thing. So in 5 years, anything can happen. How you can contain the thing, final verification, if they say, so you have more microbes, the mission will not fly. Okay, this is a complicated slide. You collect the sample from space station and bring them back. Okay, various different ways you collect the samples and then bring back to your institution and then do this culture-based analysis. Okay? No, wait. In a culture-based analysis, there is a lag between here and there. Okay? And also, you extract the DNA, okay, using some platform, and then do the sophisticated DNA-based analysis. More importantly, you cannot go every time. So you keep some samples, extracted DNA, with you, because, as I already told you, 10 years from now you will have a different technology. 10 years back, your sample come back, and then if you are using a different technology, you have to compare with the same technology, otherwise you will get confused to what you have taken with you, right? So you archive the samples and leave it for some time. And I want to give you an example of the culture-based analysis. So we need food in space. So for that we try to grow them. This is called veggie project. Okay? And also we have the environmental monitoring. So what happened, in a veggie project, the plant get fungal contamination. That means you cannot eat that. Visible contamination. Okay? So now I also got our dining table also we have measured, and we got the same fungus in the dining table. So the question comes, chicken comes first or egg comes first? Whether the environmental microbe got into the plant or the plant came into the environment, right? So this particular plant, after this happened in 2015 December, and my sample we collected May 2016. Because I collected later, that doesn't mean that came from the plant. It might have been there that get contaminated the plant, right? So the astronauts took this plant and then cut them in the dining table before sending it, because dining table, when they are not using it for dining purpose, they use it for experimental purpose. You saw that was not look like dining table, right? All these velcros are there, so that means they use that for assembling to eat the food, as well as when it is not being used for dining, they will also use it for experimental purpose. So one of the experiments, they took this and then cut those plant tissues into small thing and send it back to the earth. So it might have been done that way. That's why the spores of the Fusarium axisporum was there. But are they all the same strain? Is there a contamination problem on space station? Are projects related to microbes for whatever reason not being contained from crew living environment? This is a kind of Sherlock Holmes story. How you do? genomes are done. It is around only 60 megabase. And then my surface as well as the zinnia plant isolate were generated, and then we published that, and then look into more details about it. So some of the cucumber strain, tomato strain, and banana strain, which are all Fusarium oxysporum. But the one that is in violet color here are the space station strain. Okay, so we compared with several different genes and other things. So this space station surface strain that's from my our environmental monitoring. This is from plant project, and also we have flown Chernobyl strain to space station for various different purposes. Okay, those are all coming closer. Not my environmental surface strain, but this is coming closer. But all these things have one common phenomenon. One of them is actually isolated after chemotherapy, which is the radiation. So the common for all these things is radiation resistance. But the results are telling that my strains are different from the leaf strains. That's a good thing. The problem right now is the pathogenic strains are for plant is different from human. Okay, are these environmental strain pathogenic to human or not? Which is another issue we have to do. We use the C. elegans model, and then it actually exhibited more lethality. 60% of the worms died in 40 hours of exposure than other strain. But both of them are actually pathogenic. And then these are the C. elegans based analysis. This tells you that the time taken to kill more number of worms are really problematic. The red one are problematic. Okay. When you have a closer look, it is actually hyphae protrude through the C. elegans and then make them die. So that is the one coming from here. These are the hyphae coming out of the C. elegans. Okay, so chemistry might solve the mystery, but you guys can think about why this Fusarium produce what kind of metabolite that actually kill the worms and other things. That's one thing that you can. I have the strain that you can also test in a different way. The other one is biofilm, which I told. I already mentioned to some of you that space suits are needed when the astronauts walking outside the space to fix some things. It is a spacewalk, right? Those spacesuits are not laundered. You have to use it if your size is XL or L or M or S. It is not specifically said it is Dr. Rekha's, spacesuit. Somebody else goes, then they have to use the previous astronauts used one. That means it leave behind some of our own microbes, as you know. So this is actually spacecraft, a spacesuit material, fabric, and where Rhodotorula is forming these polysaccharides, EPS. So this is another thing that Rekha's lab is also doing. These polysaccharides can be, purified and then look at why it is forming. Okay, as you see more closely, some of them are really different shape. It produces different materials compared to the clean and round yeast. Okay, this is Rhodotorula actually, and then that are the thing. Those are these spine kind of things that is nothing but EPS, extracellular polysaccharide. Those are the things form those strings that I have shown you before. Now we started looking into the coating. So this is a small business are developing some coating for even cloth, cotton, and because it was developed for army thing. What that means? So the army people who are going and then living there for quite long period of time, they do not have the laundry facility as well. So they have to use this kind of antimicrobial coated clothes, especially the intimate thing, underwears. So these are coated with those things, okay? So that is what this panel is coated, this panel not coated. So this is Pseudomonas aeruginosa, and these are the coated material, so it get killed, complete kill. But it is not working for, Rhodotorula mucilagineusa. That means there is no one shoe to fit all sizes. You have to have multiple different things. This is for, and also material compatibility is an issue. The first one was stainless steel, the second one is Teflon. Okay? Because these materials are used to develop water processing system. In the water processing system, you have different materials. So you want to look into the microbial growth in different addition into various different materials, right? So this one is Teflon. As you see, Pseudomonas aeruginosa get completely killed, not Rhodotorula yeast. Next one is inconel, which is another material, same, get killed, but not Rhodotorula. So that means we have something working for, prokaryote, not for eukaryote yet. Now you can test these things with your, suite of different antimicrobial to let us know that, something is working and it is not forming the biofilm. That's good. So this is telling you that these are the things that allow it to adhere into the surface because of the initial monolayer from their additional materials joined in there. And if you are going to grow this with the Pseudomonas is not get killed, because the surface, first this Rhodotorula get settled, on top of it other microbes come in. Okay? So while we are measuring these kind of things, then we'll come to know novel microbes. So when I got some thing working on this, high efficiency particulate arrestance filter, which is HEPA filter, that even this facility may have something, the air circulated. Likewise, we have it in space station to get you the better air for the astronauts to breathe, right? So those filters kept there for 40 plus months, brought back to the earth, and still I have, and Asif is going to do that for some bacteriophage work. So those things actually got a lot of microbes. So I got some microbes that are yet to be described, and I named it after my imaginary mentor, which is Abdul Kalam. So I named it after him. Here is very important. We are not going to name each and everything that come across. It has to do some functionally important. It has to have something important functionally. It has to have some significance, like whether it is forming any kind of disease, like you can test the virulence, and then whether it is resistant to antimicrobial agents, or it possesses something different physical properties. In this case, this forms exosporium. Some of you may know anthracis, Bacillus anthracis, that consists of this thing called exosporium that you are not able to read here, but this is additional layer. The dark layer that you are seeing here, that is the spore coat. The one that is on the top is exosporium. You can strip off the exosporium and still the spore will work. Think like about a bed sheet that you will have when you have the cold time. So it will have additional layer to protect from radiation. So we stripped out and then show that this is actually helping this spore to survive longer period of time with this coat. So it consists of lipoprotein. So there is another biotechnologically important thing that you guys can work on it. Okay, so this is forming round spores with that extra layer. You see that this is not quorum sensing. This is because it is a adhesive nature that connects each other. and the mother cell is still, like dark here. The one bright one are the spores. The spores are like tennis racket, and then from there it will get released, and then if you further purify this SEM, so it is a round spore former, then these are, so this is Solibacillus calamii. Bless you. Then other one we found, totally around 50 plus novel bacteria we have found. I'm going to show only a couple of them. So one of them is Methylobacterium ajmalii. So this one published, and this is the rod-shaped bacteria, okay, and it actually helped to produce the hormone that is good for plant growth. So you take it to space, you cannot take tons of soil along with you to grow plants up there. You have to make use of the soil that you are seeing up in space. Like if you go to lunar thing, then have the human habitat there, so you need to grow some of the plants up there, right? If you go to Mars, same thing. So that means you have to make use of those soil. But do we know that soil is good for the growing plant? No. Even we do not know how to grow the plants in Sahara Desert or in Atacama Desert, right? So the nutrients are different. The things are different. But there you don't need to go and grow, because you are getting it. It's just a phone call away. Swiggy will bring you all things. But it is not like that there. You have to grow them there, so that means you have to come up with a different idea, get help from microbial partners, and then see what it grows, because you will be able to grow them if you have a seed with you, right? Seed means microbial seed. Lyophilized culture, you can take it with you, and from there you can propagate to the level you want, or you can extract material out, and then that can be used to enrich the soil, fix the nitrogen, all the kind of things that is required, and allow the phosphate to be, feed into this. So that is the kind of thing it produced. That's why I named it after my good friend and my senior in my class, about 7 years senior in Center for Advanced Study in Marine Biology, Dr. Ajmal Khan. The other one is, I was talking with Bhagwan or somebody who is doing the AMR thing. I don't know, Bhagwan. So he is doing the AMR study, looking for novel microbes that withstand the antimicrobial compounds. To measure fungus, you need to contain bacteria. For that you use normally chloramphenicol. Sometimes you have bacteria also grow, but it may be a very slow grower. So we got accidentally this bacteria grown in the presence of chloramphenicol, actually enriched 24 hours in a liquid form, not in the culture form, not in the agar, enriched in a liquid form for 24 hours, and then grow them. It grew, and then we got this, and named after an astronaut. I'll tell you who that is. It is also forming biofilm. Anybody knows this person? How many of you working on genomes? Just raise your hand. How many of you working on gene-based analysis? Come on, Dr. Rekha's students are working, right? Only 4 people are showing that. Don't feel shy. How many of you doing PCR? Yeah, more. I'm not asking how many of you know PCR, because everybody tested PCR for corona, I'm sure. Okay, anyway, she is the first human who sequenced in space. Kate Rubins. So she is a very good friend of mine, and I named that bacteria after her. That's why it is called Kiniococcus rubinsiae. Her name Kate Rubins. So it is not only bacteria. Fungus also produce biofilm. So these are the biofilm that I don't know why you guys are not seeing very well. So these, cotton-like structure, those are the, biofilm EPS, and these are the fungus hyphae. The dark black spots are the conidial spore. In fungus, you don't call it a spore, bacterial spore. In fungus, it is called conidia. Okay, so this we published, and then as a novel species after one of my colleague, Thomas Torok. He is a person who collected fungi from Chernobyl accident. Not now. Chernobyl, you guys may know now, Ukraine. In 1984-5 time period, there was a accident, unintentional accident, and then that is the biggest disaster of the nuclear man-made, human-made, disaster. And then he collected around 5,000 plus strain from there, the black fungi that formed, and I have all the collection, 2,500 strains of Chernobyl strains are with me. If anybody interested, let me know. So other one is Titan forming cells. Okay, what is that Titan forming? Titan, as you know, it is big cells. Okay, so Titan forming cells, that is also novel. Anybody working on Cryptococcus? Maybe no. The Cryptococcus neoformans is one of the deadly fungal pathogen that is very, very fatal. So luckily we didn't get that Cryptococcus. Initially we thought these are Cryptococcus is a problematic thing, but luckily the Cryptococcus went through phylogenetic reclassification, and then it became Naganishia, which is non-pathogenic. Okay, that's a good thing. But it's named after a philanthropist who spent a couple of $1 million for the science, Tulchinskii. And the thing is, under normal gravity condition, this is yeast-like structures, okay? Normal, budding cells are there. Even under 5% CO2 in the normal gravity, it is like this. It is a little bit bigger, but still it is fine. A kind of small stress. If you combine the microgravity, which is a big stress, it becomes ballooning. Same thing, same type, same time, everything. It balloon 5 times bigger. Why? Under microgravity, it has to settle. It produces some lipids so that it can float. If it get attached, there's a different story. If it is not get attached, it has to, otherwise it will die, right? So this one we are trying to see what, why, etc. The previous one was SEM picture. This is TEM pictures. Same size. This one is under regular gravity. This is microgravity. Even it is not fitting that particular image. Even it is not fitting there, right? So this F is same, all of them same magnification. This is under regular gravity, here is big, so you can see that nucleus there, but so many, the white dots, they are actually lipidosomes. Okay? How do you generate microgravity in the ground? Obviously, if you have money and unlimited resources, and have money to pay the $2 million per hour for astronaut time, that's a different story. But you have to first do groundwork. In the ground, you can do to some extent the simulated microgravity. So this is called rotating vessel, which is HARV. So continuous rotation, it will not settle. Okay, that is, keep that in suspension. Other one is called gravity, which is 3D. It rotates everywhere. Okay, not only one directional, it is 3D, 3 directional. And another 3 directional is this one. You can put a a big reactor also to make it, because sometimes you need to culture more. Okay, so this is the one. I'm showing you, but it is lagging. So this is the gravity that you can put 96 well plates, or you can put, even a 1 liter bottle, everything in there, so it rotates all the places. Then you can measure them later whenever you want, whatever you want. Okay, so we had multitude of omics done. Okay, genomics, proteomics, lipidomics, metabolomics, in addition to the culturing, everything, to measure what they are. Okay, obviously it is lagging here to there. Obviously, growth in this particular case, because it is growing bigger, so growth is a little bit less in the microgravity. If you are growing at 22 degree, it is equal, but if you are growing at 30 degree, it is not. And the cells are bigger. Obviously, you see all these things, lipidosomes I was talking about. And when you do the lipid analysis, it is entirely different. Lipidomics, this is entirely producing different compared to the simulated microgravity versus regular gravity. What they are, this is the volcanoplat, okay, and then you see differential expression there, and then when you take a closer look, it produces prostaglandin, which is actually in wound infection, in the fungal wound infection it causes. So some of you are working on wound infections and other things, so it may be a good model to work on why it is producing this. Okay, this produces 4-fold higher prostaglandin under simulated microgravity condition compared to the regular thing. Okay, it is presumed that this novel fungus might exhibit enhanced virulence property under microgravity. To confirm, we need in vivo studies using murine model or something. The other one is proteomics. Also we did, as you see these different quadrants, you see the different kind of things expressed. This is simulated microgravity, this side of 30 degree versus 20 degree. So entirely different profiles you are seeing. And what are those proteins regulated under microgravity in this particular yeast? Okay, some of them, as you see here, expressed. This is the one that you have to focus on, what it is expressed more. Similar, something expressed less. Okay, ricin B like lectins have been shown to possess immunomodulatory properties. It exhibits anti-proliferate effects specific to human leukemic T cells. And other ACADs, which is a dehydrogenase, and flavoproteins are involved in beta oxidation of fatty acids. So fatty acid oxidation is a primary means of producing energy supplied by oxidative phosphorylation rather than glycolysis. So those are expressed higher. And some are not. Some, like unsaturated fatty acids, are down-regulated. So in this case, that some desaturases that are the key enzymes for the biosynthesis of unsaturated fatty acids are down-regulated. So these are some of the omic studies, influence of microgravity on them that I am showing you. And some of them are persisting. Some microbes are persisting. This also I talked with Dr. Rekha's group yesterday, and what chart you are seeing here is only BSL-2 microbes. This is called word chart, which you are not able to see and read. I don't know why. Maybe it is good in YouTube, I think. The bigger the font, that means more number of genes. This is based on metagenome. Okay, so the one that is in violet color are Klebsiella pneumoniae. Okay, so it started from these are the flights. Flight 1, 2, 3. 1 and 2 have 3 months difference, and then after that went back. Sorry. After that second flight, we planned for the Come on. We planned for the third flight, okay? Third flight did not fly because SpaceX 6. Anybody know what fate of SpaceX 6? Come on, it died in the sense we had a mishap. So we have to wait for 1 year for the next flight to come. Okay? So that is what this is. So flight 1 and 2, 3 months, and the 2 to 3 is 1 year. Okay? So we found a strain from location 5. These are all locations defined. Every time they go and then collect, we have a market, demarked where to collect. Even the astronauts leave, the other astronauts come and collect the same thing. So we are doing it for nine consecutive flights. Okay. So location 5, we are continuously seeing all the time 3 flights, and then in the third flight, it actually spread to other locations. So this is clearly telling us that there is some problematic issue out there. This, even though the astronauts are asked to maintain the places, they can do only so much. Think about yourself. If you are asking during Corona time, let's take that, you don't have a maid coming into your place. You have to clean your bathroom by yourself. Entirely different. You clean yourself is different from you get a very, very good janitors that you hire, right? That's how it is. Maintenance is a big, big issue. Okay. So the second strain is actually from location number 2, which is the space toilet. Okay. That's where Columella also found. And then we want to see whether these 2 strains are same or not. Is the spread of Columella species an opportunistic pathogen coming from one strain? If so, how to find it? Metagenome analysis can be carried out to source track the strain, specific traits of the Klebsiella pneumoniae. We have the data in a specific place called Gene Lab, NASA Gene Lab. Then you can mine them to know whether is it only one single Klebsiella strain or what. If so, how to eradicate? This is where ASIF is going to do it, find a phage to see whether that can be eradicated. You see, if we are successful, we have a strain. If we are successful, we can mix with the cleaning reagent and use it in space. Next one is microbial warfare. I'm just passing and to see if anybody understand what is this. Microbial warfare is nothing but like a biological warfare that people are using it in the battlefield, and the microbial warfare is nothing but a bacteria or fungi fight for its niche, not allowing others to come. Okay? How do you do that? You can do it with the metabolic modeling. Okay? So this is the work that I was talking with Rekha the other day, that IIT Madras person is working with me, computational scientist. Anybody computational scientists who are interested, this kind of work is very, very important. So you first model metabolically and then prove experimentally later. A lot of people in the DNA era forgetting to validate your prediction. Everybody interested to predict, but you need to validate your prediction using experiment. Okay? So this one is, we collect samples, as I said, 8 different locations, 3 different times, and then they did all this modeling work, and then which one is actually connect to each other, which microbe is connect to each other, which presence is necessary for the other one. Obviously, some of them are good presence, they are mutualistic. Some of them are symbiotic, but some of them are parasitic. That's what we want to know. The parasitic thing that you are seeing here, this is the parasitism. If the mark goes from one side to the other side is not, that means it takes advantage of the other one and then kill them. Okay? So the Klebsiella pneumonia, most of them are okay, and not okay. Most of them it actually kill, and one get killed by other species. Rhodotorula JG1B is actually acting up on this one. So that means we have both we can predict here. So this Klebsiella is producing antifungal compound. Okay, let's prove that. Right? So this is the SEM picture. This is the SEM picture. In a 4-quadrant. On the left here is the Aspergillus fumigatus, which we found from space station, which is a BSL-2. It is a pulmonary infection it does, okay, like pneumonia. And this under the regular growth, regular condition, normal gravity condition, it forms a beautiful flowerette. This is called conidiophore, and these are the conidia. In one single thing you have thousands of things, just like a flower. Think about a flower. Okay, it forms like that way. But if you add this red thing, which are artificially colored, the Klebsiella pneumonia, into that and grow them together under regular gravity condition, it produces something, and it started killing. The spores are not formed here. The formed spores are get eaten away, grazing them. Okay, but if you grow them under the simulated microgravity alone, still it is a stress. This is what difference. You are healthy, but if you are having some fever or something, you are immunocompromised. That's where you have other things get attacked, other microbes. So similarly, under the simulated microgravity, it is compromising something. It is, to some extent, it is immunocompromised. When you do the growth along with the Klebsiella, completely bald. And we proved both by culturing them, number of fungal cells in here versus here is completely less. So now we can take advantage of Klebsiella pneumonia, even though it is a pathogen, find a metabolome of it and look for antiviral compound, antifungal compound that can be used against the fungus. Now come to the main story of why I was connected with this institution and Dr. Rekha's group. Metagenome to phenome. Metagenome is nothing but a shotgun analysis that you do not know what gene. You don't need any kind of a priori information. Something like you shoot and the target is there, right? But the target may not be there. You shoot 100 times. Still, if it is not a shoot 1,000 times, you will definitely hit the target. That's called shotgun metagenome sequencing. So now you have the genes. Now you can align them. All the 1,000 times, whatever you are getting it, now you can make a strand of genome. Now you complete the genome. Now we started getting this red thing that you are not able to see there. That is not described at all. If you are not describing, you cannot name the bacteria nowadays. You have to culture them. Now with the metagenome analysis, now I have already archived 6,000 strains in my lab. So my team went back and then looked for this specific gene, and screen all 6,000. Boom, you got some cultures. That is what in NIH and everywhere is there as an emerging pathogen. If they cannot identify today, they leave it as an emerging pathogen. Same thing for our coronavirus. Was it came from God? Does it come from Mars? Everybody say it might have come from Wuhan area, but there's a different story. But it was there. Our technology didn't know and could not identify such a mutations. And it found its own niche and started spreading. Okay? Likewise, we found this from our culture collection and then published in 2018. Poor Rekha was having this thing but did not publish. And we published after naming my, virtual mentor, Kalam. So I named it as a Kalamiella as a genus name. Before I showed you Kalamiae, which is the species name. Some of the microbiologists know what the genus is, what species is. And then one of my colleague from Johnson Space Center, he retired, and I named after him Pearsonii. His name is Dwayne Pearson. He's also famous space microbiologist, pioneer in US on the space microbiology. I named after him because one of the 20 full metagenome assembled genomes, which is MAG, retrieved from the ISS environmental metagenomic sequences, were the one that we were able to show, and also single cell assembled genomes are also done. And then culture, metagenome, and single cell genomics approaches yielded such kind of results, and then become this. So now I got this Kalamiella, and then I was reading some paper from here. There was written like first report of pathogenic bacterium. We got stunned. We thought Kalamiella is not a pathogen because ref finder, which is this to genomically predict, you have a algorithm to look into the virulent genes only. That did not show us this is virulent. That did not show us like Kalamiella, Klebsiella, pneumonia, and Enterobacter buchanensis. These are the ones that we found from space station. They're more predicted as more resistant as pathogen. But this did not. But then came the uropathogenic thing from Rekha's group. Then I contacted her. Because she is also very much interested to get connected with us during that time, and this is her paper that she actually synthetically grow them, and crystallization of the Kalamiella pearsonii was done by her group. Okay, this actually made us to know more about these things. And then while we are working on developing the collaboration last year, we got this news came out. Human disease caused by Kalamiella pearsonii from New York Hospital. They published in 2022. This was a 56-year-old woman with a medical history of end stage of non-ischemic cardiomyopathy. But it was treated after 14 days of intravenous piperacillin tazobactam, which are the antimicrobials treated. But that was actually eye-opener for our group. Still NASA is puzzled about this bacterium that we are getting again and again and again for the past 9 times. It might have gone through human, the crew up there, and then contaminated, and we are not able to clean it. And then the Irish group contacted me. He is the one right now doing, he resequenced all my strains. He is going to resequence Rekha's strain. He is going to resequence the strain from New York, and also try to do the murine study. That means, do the virulence using the animal model. Okay? Because they found in their hospital oral microbiome. They are really scared now, because if they get the second one, then it will be a problematic for their hospitals. They have to clean the hospital so much. Okay? Just before I left the US to here, I got this call saying that they found it in German hospital in University of Cologne. So these are also being collected right now, and all those things will be given to Rekha also to do the urease analysis later. But first, it will go to the Irish person, and I gave all my 20 different Kalamiella pearsonii I found so far from space station. So he is doing this. So it is very important. This is the Kalamela story that I have connected with, Rekha, and my presence today is because of that story. Okay, so I told you a few things, and in the interest of time, I'm going to touch only this omics in space here. I think I'm not boring you guys, and this instrumentation thing will be very interesting, at least to me. And there is a video. Hope this will come out with a good audio. For the past couple of decades, microbial monitoring is happening based on omics technology.
Omics includes everything inside the microorganism, not just their species identification through their DNA, but also what enzymes they're producing, what proteins they're producing, and what metabolic they're doing. And as we study microbes in space, these are key things to take into account, because those can give us clues and hints as to how microbes are changing and adapting to the space environment.
So currently on the International Space Station, there's no mechanism to be able to process samples. The process for microbial monitoring on the International Space Station is for astronauts to collect samples from either surfaces or the air with swabs. Once the samples are ready to be returned to Earth, it gets put into a capsule and then it lands into the Pacific Ocean. A boat then comes to collect the samples, which is then transferred back to different NASA centers. So it's very important that if we could cut all of these time and these losses by performing sample processing on the International Space Station itself.
So one of the things is we need an automated sample processing instrument, which we call them as SPI system, that will be helpful to measure the microbial population in space in a closed system during the long travel.
The SPI is a microgravity optimized DNA extraction device that's ideal for use on the ISS. The crew time will only spend about 1 to 2 minutes per sample, and then they could use our device to process multiple samples simultaneously, which means the data that they collect eventually will be more reliable, simply because more sample and more repeats can be performed with our device.
So once an astronaut collects the samples, it would take only a day for us to get results back rather than about 4 months, which is now the current protocol.
But we do not know that it is going to work in space because fluids behave differently under microgravity.
The SPI device can successfully extract DNA on the Earth's surface. However, the fluid properties of liquids in microgravity environments require novel engineering approaches. Specifically, a new well-shaped design will be required for the SPI to function in microgravity.
So if you don't design for microgravity, you can have fluids in all sorts of places. You can have the liquid on the top, you can have it on the side, you can have it on the bottom, you can have droplets ejecting, you can have geysers created, and you can have all sorts of things where you really just don't understand what your fluid's doing or where it's going, and it can be very dynamic. And in this project, what we're doing is we are taking a closer look at the well plate design, and we're modifying that so that it works predictably and efficiently in a microgravity environment. In a system that's designed for microgravity, you'll see the fluid at the base of the container, and it'll be just like you're in 1G. It'll seem like gravity is actually present because all of it will go to this well at the bottom. And so you create a predictable, simple system that's passive and is self-correcting.
JPL and collaborators developed a pipeline, a microgravity independent pipeline,
There is a sequence
starting at the species, extracting their DNA, prepping the DNA, make it ready for the sequencing step, sequencing it on the biological sequencer, the Oxford Nanoparmenium, and actually be able to analyze the data on site, on the space station.
The data that we receive from the MinION can then be sent to various bioinformatics pipelines.
We take those files and process it as per the codes and open source software that are available to us. We do a quality check analysis. Each and every sample is checked whether we got the right results. We check it through the rare fraction charts, and the rare fraction chart tells you what type of assembly you will get and what type of organism or diversity you will get out of it. Once we have the quality reads with us, we do the identification and function analysis. The function analysis tells us what the system itself is doing here, which tells us what type of organisms are there in the sample or what type of processes they are doing.
Now that we have a pipeline put in place where we have DNA extraction on the SPI, sequencing on the mid-ion and bioinformatic analysis, we had to put this pipeline to test. We decided to test this in a variety of environments that fluctuate from low biomass here at JPL at a clean room and high biomass in Yellowstone National Park on multiple biofilms that form in hot springs.
The hot springs of Yellowstone National Park provide an ideal environment for studying microbial ecology because they're remote, they have high biomass, and they are extreme environments. And this is analogous to how the SPI device will work on the ISS. It's a remote environment as well.
And what's so great about this test we performed at Yellowstone is we started getting data generated while still in the park. And before we even left the exit, we started getting sequence calls.
The data we generate in the field from the samples we collected there looks very similar to the similar samples that were processed in a laboratory environment, with the caveat that we were receiving our data almost real time versus days to months later.
This demonstrates that the SPI mid-ion bioinformatic pipeline we've developed here functions in remote settings.
So with this miniaturization technology, we will be able to analyze microbial population dynamics in space station. And perhaps even in a future crew vehicle that would go beyond the moon and Mars. All these people that you have seen are my group people, either postdocs or scientists there working. So when you are developing such kind of technology, think about you are giving blood samples to for your regular checkup. The doctor will say, come later 3 days, then I'll do it. You will get the results, even in the earth now, right? Think a system there right now next to the doctor's office, then he can prescribe the medicine. Until then, he will give you a generic medicine to you. But if he comes to know that something is really a specific thing, then he can tune his treatment differently, right? So likewise, and also similarly, we wanted to first extract the DNA. To extract the DNA to get this machine, it has to be miniaturized, right? And it has been tested in the clean room, as you have seen, and it has been tested in the Yellowstone. It has been tested in the back of the vehicle, which is a vibrant thing, 85 miles per hour, which is 140 kilometers per hour. You can do it in India, okay? But in US, so you run on the back of the car and then see how it is working with the vibration. Everything worked fine. So we published this paper. But important thing is microgravity. Is it going to work? Because you saw what the problem in the microgravity. Fluids will fly everywhere. One of the astronauts who was from Italy could not bear drinking coffee in the Tetra Paks. He wants an open cup. So he threw this idea, I want an open cup. So some material scientist people designed that coffee cup that you have seen. So astronauts can sip the coffee in an open cup now, because even upside down it is not going to spill because of the property of the material. So same materials we are using it. It's different design, but same material we are using it for our purpose. Okay? Now, in the ground, you have to test the microgravity. There is nothing you can simulate microgravity here, other than this parabolic flight. What is it parabolic flight does? 20 seconds of weightlessness you can get. So if you have 1.5 or 2 hours flight, every 20 seconds you get 20 seconds, and go up, and then come 20 seconds, so 40 times you'll get, so you will be able to get 2 to 3 minutes of your time. So every single time you can test one and test second. This is important before you fly. What I am trying to tell you, these are the challenges we have. It is not that I found something, some electrochemical properties done here on the earth. Is that fluidics that you have can be measured appropriately, can be contained appropriately. Otherwise, you are going to contaminate the station. So this is my postdoc, Seth Parker. And then if you take the current microbial monitoring system, you collect this swabs or samples and store it, and then you saw that how it is being dumped into the water, and then all these things take around 4 to 6 months to measure what it is. If you have a DNA extraction system that is working under space, all these things can happen within few hours. The time taken here will be roughly 9 hours to get the same thing. But first let us test whether it works under parabolic conditions. So around 6,000 meters, the NASA flight, NASA aircraft fly, and then up to 8,500 meters it goes, within few seconds, 20 seconds, you will get a hypergravity. Then you will get weightlessness, 20 seconds, like that. Okay? So under 20 seconds, this is a real picture. My postdoc's picture is there. And then this will give you an idea what will happen under the microgravity. Most of you know that pipetting, and then all the pipettes are flying everywhere if you are not containing gravity. So fluid transfer has to happen in a contained form, otherwise this is the one you are going to happen in space. Okay? So we took it into this 0G, and this is the instrument that is getting into there, and then we are loading the cartridges. This is called microtitan, a sample processing instrument. We load it. All those things are contained in the surface of the 0G flight. Okay. And then this is the flight pattern. What are they doing? Simply jumping and down? No. This is 20 seconds. They are experiencing that weightlessness. This is called vomit comet. Everybody vomit. All this flight is every time after that they clean thoroughly. Otherwise you will get only vomit things around. But anyway, so you see those spikes, that is when you get the weightlessness. It is only 20 seconds. With that time you have to do your experiment. It's not only your experiments going on. You have 50 different experiments going on at the same time. Okay, this is the successful thing. So he gets strapped on the floor. Okay, that guy, see that guy gets strapped, and then soon you will get this weightlessness. So now they get weightlessness, and he is observing what is happening in that instrument. The guy on the left side is a very experienced. He is swimming nicely. All of them are falling down, upside down. They are first time riders.
Yes!
Yes! And then he said it is nicely moved from one well to the other well. Okay, that you have already seen. Instead of pipetting, automatically the things are moving from one well to the other well. Okay? So with this, in the interest of time, I'm not going to go in details about several things. This is not one-man show. This is the combination of several people over the period of time, 25 years worth of my thing, which I have shown only 2 to 3% of my research here. But this was possible because of these people, and next time when I'm talking, Rekha's photo will be there in it. Thank you very much. And one more thing is, you guys should ask question. No question is dumb question. Anything you can ask, even non-science nonsense question is also fine. Especially I want to hear something from the girls.
Yes, it was a wonderful session. I can say it was like an encyclopedia which we all were curious, and it was like a gist of the visualization what happens actually in a spacecraft, or how the samples are collected or processed and omics happens. I guess all were more excited to see the videos which are shown at the last slides. So the session is open for discussion now. Anyone, any kind of question, as sir told, he is ready to answer. Take this opportunity and you can ask any kind of questions.
Go ahead. Take the mic.
It was very interesting to see the people floating around under microgravity. I work with stem cells. I have read some of the papers in which they are showing hypertrophied cardiomyocytes under simulated microgravity condition, and then the cultures have been kept in space stations as well. So my concern is that how about the health condition of the people, like they are very youngsters, I guess, and do they have chances that they get some cardiac abnormalities or any kind of abnormalities working in these kind of conditions? How is that ensured?
You are talking about the crew and astronaut, right?
Yeah.
Yeah. So number one, astronauts are very, very, very healthy person only we are getting it. Okay? Very healthy. They go to rigorous training and all. It is not easy to, get into that, crop. Number one is very, very healthy, but that doesn't mean they are not immune compromised. So one thing that I can tell you, which is fully published, is viral reactivation. What happened? L minus 6, launch is L, minus means 6 days before the launch, okay, L minus 6. The crew, they are only 40, 45 years old. They should not get shingles. They should not get herpes virus. But herpes virus and shingles get started into our system, who are 55 and 60 years old, get resurfaced because at that age we are stressed. The astronauts get stressed, however much they have flown before, they get stressed because this flight is coming, which is very dangerous for life. Very dangerous for life. Because of the stressful condition, the virus, herpes virus, until then is latent, get reactivated. You get more tighter. Once they go and then land, and if they are going to be there for 6 months, it is now become normal. And then now, return -5, now they are coming back, again stress level high, and then herpes virus tighter is more, but once they land here it is fine. So the stress is important. So however healthy they are stressed. But your question is, do they have experienced any cardiac disease and other things? As far as we know, there is no big thing rather than the rashes and other things that they had, and some had some, eye, defect because of the radiation, and they have not reported about the cardiac, heart-related disease. And one more thing is, even though you are in YouTube, but I'll tell you, the astronauts who experience certain things like this, may not reveal. That's my thinking. They may not reveal because they need to go again. And their health and medical history is protected. They don't need to reveal their medical health and the defects. Okay, so same thing, you go to, you are getting corona, you want to leave, and then you want to get into the flight, and some people are not going to reveal it. But there is no major health problem, for sure. Otherwise, that leads to cancerous thing and other things, will be, revealed and will be in the news, but it is not there. They are very healthy.
Thank you.
But 2 years to Mars, we don't know. How about students? Students need to ask questions. Yeah. For one here and then second one is there.
Hello sir, it was a wonderful presentation and it was interesting too. And my question is, I'm just
keep the mic next to your mouth, yeah.
Yeah, I am interested to know, you mentioned about the identification of samples from moon. So what were the sample? You were able to collect soil sample or something from the moon?
Oh yeah. Rock, soil, everything.
Okay.
It was there during Apollo days, yeah.
And you didn't identify any microbes in?
No.
Okay. Thank you, sir.
Yeah. Someone there? Maybe it is better to line up here.
Hello, sir. Sir, in one of your video, I saw that the tips were falling that side and this side. So my question is, while in space, to grow organism, we need media, right? So we are pouring in the space, or we have to take from here to there?
We are not growing open petri dish. Everything is contained. So what we are doing right now is more fluidics is going in and then get filtered in a contained area. After that, the another fluid is injected. Everything is either manual injection, which is connected. It's just press, okay, and then it goes in there, and then allow it to grow. And it is getting more concern. If you grow bacteria in space or microbe in space, is more problematic than leave it alone. In other words, if you have one E. coli, that is not going to harm you. But if you are going to grow them, one E. coli become colony, and you are having artificially inflating to billions of cells, that is a biological bomb actually. You should not grow things in a contained place. So that's why NASA is coming out of the culture-based analysis to molecular-based analysis. For that, these kind of capabilities are coming. We already shown that DNA and other things get sequenced in space. It's possible, but what is not there yet is sample processing. You collect samples robotically, not there. Once you collected them, how do you process them without getting these kind of fluidics issue? So that's what we are developing. Once it is complete, then sampling to sequencing will be happening. So to answer your question, we are not growing in space. For that you don't need to make any media there, but we have a contained microbial measurement system that is being used.
Thank you, sir.
Yes. Here? Let's get up. If you get up, they'll get.
Hello, sir. So
Yeah.
I just want to ask that what sort of research is happening in the astrobiology area in the NASA? And because this is my first question. Can you give me just broad perspective on that? What sort of experiments are happening in astrobiology domain?
Okay, thank you. Which I have not touched more here. I told you in the beginning that we know Earth-centric biology right now. If you use the Earth-centric biology, you cannot find indigenous life of Mars. So you need to come up with the prebiotic mentality before amino acid, how it is amino acids formed. You need to have astrophysics and astrochemistry in your mind to get the astrobiology going. Okay, and exploring in Mars, we are also trying to look at the thing whether anything extinct happened. What happened there, the biomolecule wise, what happened? Is it chirality of the amino acids different from what we know on the Earth? Okay, so mainly astrobiology focuses are on biomolecule detection, how it is formed in the before biology time, prebiotic time. That's why we are going into different extreme places. We go to hydrothermal vent, which is 420 degrees centigrade. If you think about it, DNA cannot withstand that. It will disintegrate. But still, now we know 113 degree centigrade archaea can multiply. Caltech found that. When during the time that 1977 time period when hydrothermal vent was also found. Unlucky to NASA that already sent the Viking mission to Mars by baking the the spacecraft to 112 degree for 96 hours. It totally take the thing that I showed you, the lander, put it in a big oven, which cost $200 million that time, okay? But now you cannot do that because you have composites and other things are different. So what I'm trying to say is, every day we come to know new things in the biology by exploring our own Mother Earth in extreme places. That is also helping astrobiology to what to look for. So those kind of things are going on. Did I answer your question?
Yes, sir. That means, like, are you focusing on the primordial soup, basically?
Correct.
Starting from the RNA part?
Correct.
Second question is about, like, what is the effect of the microgravity on those stem cells? Particularly, I came across that the astronauts face some issues in the bone, something like some degeneration, mostly. Any study that's been done?
If I understand your question right, there are two things. Astrobiology is mostly robotic missions right now. It is not human mission at all right now. Okay. Tomorrow may be different. Developing a sophisticated laboratory up in Mars, and those things will come later. Maybe it takes 50 years from now. I do not know. But if the growth of the instrumentation is like a Moore's law thing, going extremely good pace, there's a different question. But right now everything is robotic mission to look for life or collect the samples, bring back and look for it. So with that, the only what we know is biomolecule based detection. Nothing else we could do now.
All right, thank you.
Nobody here?
So I would like to ask about the relevance of research in microbiology in other planets besides Mars. Like, what is NASA investigating on other planets? Like, do we have a scope of researching on other planets besides Mars?
Yeah, Mars and also the moon of the Jupiter called Europa, and that is the next thing. It's called icy world. So the moon of the Jupiter, Europa, has around 2.5 miles thickness of ice. Below you have a brine solution, like think about the Antarctica have the salt water, ocean water down, but you have a thick 2 miles of ice, Lake Vostok for example. Okay, so that is another one that we are looking for, because water is very important component for life. If you don't have water, you do not know how to multiply things. Water is very, very, very important. So there is no issue of the water of the for Titan. Did I say Titan? Europa. Europa is the Jupiter moon. Okay, and the other one is Saturn moon, Titan. So these are the things that we are trying to explore next. After Mars is trying to land in Europa, which is a big, big thing, which we already started looking into the orbiter. We have orbiter to orbiting there, but lander is the next big thing, is coming in a couple of years. Once you land there, then you come to know the geochemical and geothermal activities of this, Jupiter moon system. So those are the things happening right now.
Also, sir, Neptune, I've read about Neptune. That's very cold in Neptune. So I would like to know, is there a scope of microbes in Neptune?
We don't know.
No.
I don't say no, I don't say yes, because I'm confidently say we do not know.
Okay.
Good afternoon, sir. Considering the fact that space is an isolated area, can there be research done on organisms belonging to biosafety level 4?
You are talking about in space?
Yes.
Okay. First of all, you have to contain even the BSL-1 microbes there. You cannot contain. That's the biggest issue. So BSL-4, knowingly, they will not allow anything beyond BSL-2. If you know that it is, more than BSL-2, no. You cannot even take anthracis, which is BSL-2. Deadly microbes that are BSL-2 are not allowed because you don't want to take chance, right? So that's one thing. The other thing is we may be able to use the space station to step in first. As a test bed, we can bring the sample from Mars to the space station and analyze a lot of things before bringing it to the Earth. So you are only, compromising 5, 6 astronaut, not the whole Earth, right? So that's the thing we are doing. But we are also doing contained measurement, autonomous measurement without human, the one that is flying to beyond Moon to the Mars. So there is gateway. So those are the things happening, but everything is, by robotic missions. Everything, everything is robotically measuring, no human right now. So then we will have, whether containing this and that, then we'll have a human in it, and then see how it is happening. And then we built station in Moon. We don't have any station there. So we have to build the station where human can live without any kind of radiation effect and other things, and then see whether they can grow rice, for example, wheat. Then they learn from there then before going to Mars. These are the test beds. We already did with this International Space Station, but the rest of the things are slowly happening.
Thank you, sir.
Here.
Good afternoon, sir. My question to you is, like we have microbes on Earth, they have certain growth factors and characteristics that define what kind of microbe it is. So microbes in space, what could be the characteristics or how are they different from Earth?
Okay, there are two different things. If you are taking the space station, they are the microbes taken by us through the human, through the cargo and all those things, okay? So that we know and define what it is based on the technology that we know today. But if you're asking microbes in other planet that we do not know yet, you have to look based on the biomolecule, like I explained to that astrobiology questions there. So you need to first define the biomolecule and then know whether they are the microbial entity or not. We don't know, how Martian look like, for example. Or we are surrounded by Martian right now that we don't see the wavelength that you could see. So those are the things we don't know. A lot of don't know there when it comes to life in other planet. So, but biomolecule based studies will help us. Some people will say, hey, inside to a measurement only to some extent you can do. We have to bring the sample back down to the Earth to completely characterize. And some people will say, don't bring anything that, to kill all of us, all the humankind. Don't bring. So that's another thing. So this is catch-22, but we'll see what will happen.
So got another one for you. Now you told that we take the microbes in the space. Now, the microbes present here on Earth is adaptable to the environment and the conditions that's suitable on Earth. So, but the space is a completely different thing. It's a completely different concept. So, are these, how are they even able to sustain in space? Won't they die?
When you said space, you have to define the conditions of space. if it is inside space station, for example, human are living. Human are dumb. If human live, microbes will never die. Okay? But if you are putting it outside the space station, for example, which I have not covered, 99.9% of them die because of the radiation. It cannot withstand space vacuum. It cannot withstand space radiation. Okay? So 99.9%. Because why I am saying 0.91% is still there, because we don't have a technology to comprehensively measure everything. That's why we are collecting samples outside the space station, which were exposed 22 years now, whether anything sustained there for long period of time. But inside the space station, comfortable, because human living and electronics has to work, and all things are little bit of extra CO2 there, little bit of extra radiation there, but it is not lethal to human. So it causes some cancerous compounds, that's for sure, but those are localized, not globally kill everything. So.
Thank you, sir.
Excuse me, sir. There is one question from online person who was watching. The question is whether the new fungal species that you described had speciation events happened because of prevailing conditions in the space station. So what do you think about this?
No. The answer is no. The evolution required millions of years. Space station is only 22 years. Then why we are finding so much novel things here, right? You go back to your garden soil and then do comprehensive analysis, you will find new bugs tomorrow. Okay? The one that is stopping you not able to find is the dominant microbes on your garden soil, which is only 10% of the microbes that you know today. That forms 90% of the total microbes. One is total microbial burden, the other one is microbial diversity. Diversity is never end tail, but it may be their small percentage, but they also define the ecosystem. They also help the dominant microbes to sustain in there. So what I'm trying to say to this online person is, evolution has not happened, but adaptation and mutations are happening, which is right, when it get hit with some sort of radiation thing, yes, point mutation might have happened, or 20 plus years of things, it has to adapt. So over the generation it adapted. So space station, new microbes actually came from us. One classic example was, 2,000 meters down, a subsurface molybdenum mine, I found a bacteria. The other strain was in spacecraft assembly clean room. It is not some mine worker came and contaminated my place. Okay? So everything, everywhere, environment selects. Okay? The other one is deep sea, 11,000 meters down, the Kyrie found a bacteria. The other strain I was found in the in the spacecraft assembly clean room. So likewise, these novel bacteria and novel microbes, novel yeast, novel fungi that we found are because we are looking deep enough, and also I am selecting to some extent that the microgravity and the radiation kill the dominant benign bacteria which are not able to cope up with, will die. So the rest of the things are there when I am collecting. So those are, I have more frequently come across those novel bacteria.
Hello, sir. It was a good presentation.
Yeah.
I have a doubt, sir. In Mars, there will be a lot of microorganisms, sir, in soil and all. So
No, I didn't get it. In the Mars, a lot of bacteria inside?
Yeah, like microorganisms. Like
why you are saying that?
There will be no, sir.
Huh?
There will be a lot of bacteria and all.
In Mars?
Like in soil, like.
In Mars soil, a lot of microbes? Where did you read that?
No, sir, I'm asking about that actually.
No.
No.
The answer is no. We don't have it. As we know today, we don't have the microbes floating around in there. There may be some subsurface there which we don't know yet. That's why we need to get the sample down here. Inside to exploration for biogeochemistry performed, we did not find any biomolecule that we know of today. The probe that we sent may not be sensitive enough to do and then get the answer that we want. That's why NASA is bringing the sample back to Earth to find whether it is there or not. It's not a dumb question, don't feel that way. It is a good question. To get clarified, no microbes that we found today, Mars, that's why it is called red planet. Otherwise, by this time, it is green planet. If we have microbes up there, it would have grown, and then it become more green. So that's why it is called red planet. So it is barren. So right now there is the radiation will is taking care of everything. Anything around 220 nanometer and above are there. So what we are seeing here, 254 is DNA get modified, right? So 254 is the UV that we are actually shining to kill the microbes by disintegrating the DNA. But in Mars, you have 220 nanometer and above things, which is in the range where it's biocidal in the surface. But one thing is which I haven't shown you because today's I don't have much time. If you have a thin layer of Martian soil on your bacteria, UV doesn't do anything. But gamma is different. Gamma penetrates. UV is not.
Thank you, sir.
Good afternoon, sir. Sir, my question is regarding the biofilm formation in space. Like, are the microorganisms efficient in forming biofilm in space, just like that of Earth? If yes, what are the possible environmental stresses which they face, like regarding the change in pH or temperature or any of that reasons?
Yeah, so there are multiple questions that we do not know. We have these microbes that forms biofilm. That's why we are looking for collaborators to do the chemical biology of it. What kind of chemistry it is producing, whether it is, as you said, extracellular polysaccharide or any other, glycoprotein that it is producing. The effect is big. One is biocorrosion, okay? And clogging of the bioregenerative system. That means it forms biofilm inside the tubes, the pipes, okay, and on the back of the coolant, a refrigerator kind of thing system there. So on the back of it, you form this biofilm. It is blocking the flow of the water. So these are the things happening, but we are looking for remedies. We have mechanical abrasion, to dislodge the biofilm. We are also trying to see whether we can paint with the chemicals that is not leaching into your water systems to those are the developing the countermeasures of the biofilm formation. Still a lot of things has to be done, and this will be useful in the earth also. So think about the marine industry where they are transporting oil from in the marine industry. You have a lot of biofilm. The bivalves mollusks which attached into it, initial thing is bacteria. Bacteria form that slimy layer before this bivalve come and get attached. The larvae come and get attached and become barnacles and other things. So what I'm trying to say is we need to develop countermeasures for futuristic human habitation in space. We are working on it. So think widely, think anything, that is useful, and then that may be one of the things that these agencies can adapt. Still a lot of things to be done.
Thank you, sir.
Last question?
Good afternoon, sir. Sir, my question is like, Earth has ozone layer. Like Mars has any kind of layer that protects the radiations from sun?
No.
Then how it is possible to say that Mars can
that's a question. So that is people saying there is nothing in Mars, so why don't we go and then try to terraform them? Terraform means, take the terrestrial materials up there and then make a farm, like grow plant in there. But you need to have appropriate shield from the radiation if you are going to live there for a long period of time. No, that's very, very important. That's why human habitation in Mars require all these, physical barrier and other things. So without that, it is tough. That's why I already said, 220 nanometer and above are still coming. You don't have an effective atmosphere like what we are enjoying to protect this, radiation things that we are getting only 400 and above, not below. So UVC we are not getting. UV A and B get trapped by the layer, ozone layer that we have. Such thing is not there in Mars. That's why it is barren. That's why it is desert-like.
Thank you, sir.
It's okay. I'm not tired.
Any more questions?
Here.
Hello, sir. I'm Sumaya, doing my second year PhD. So, sir, this is like, I cannot dream beyond working in NASA now, in my second year. So, did you think, sir, doing your PhD, that you will be working in NASA in future?
Yeah, why not? Why not?
How far it was,
learning,
sir? How did you achieve it?
Learning is always there. Every single day we are learning. If somebody says, I have a PhD, I know everything, then they are lying. So there is a first step to, get into the science and then, think critically what the gaps and needs are of a given situation, okay? So anything possible.
Thank you, sir.
Yeah. Data science people.
Good afternoon, sir. It's a very nice presentation, and it's an honor for me to talk to you. And I have general questions, sir. First is what motivates you to enter in NASA after your PhD? That's already asked. And thing is.
So first question. Okay. So, what are the steps needed to get into NASA?
Yes,
okay.
Sir. Especially after PhD.
The answer is you have to be somebody. Number one. What that means is you have to be showing that you are doing something that is needed, okay? Not only PhD, the quality of things that is needed for the NASA's question. In this case, if you are having some biomolecule measurement and other things, that's one way. The other way is, the skill set that you have that is required for, NASA scientist, you can enter as a postdoc. Okay? But NASA is not an academic institution. NASA is federally funded things doing specific things, space science. Mainly because we have a good infrastructure that no one has it. No one can build a spacecraft other than NASA, right? Obviously, the companies like Boeing and other people are, able to do it with the help of NASA, right? So NASA should not compete with academic institution. So that's why we don't have an academic program, but we have an internship program, and that too mainly for, people who are living in the United States, mainly because NASA is an entity paid by the public. It's a taxpayer's money. So opening up to international thing is possible if that international students come with their own support resources, not for doing research support, for their living support. For example, if Dr. Rekha is having money through the DBT, and she can send some students to the NASA institution for short period of time, 3 months, 6 months, or even PhD program. So, living there for 1 year. That is up to a kind of, NASA scientists need that kind of skill set, has to be shown that, yeah, I want this person because I don't have, much resources to spend, but I do this research resource I can take care of, like buying things and all, but she is motivated to come with her own institution funds, then we will help.
Thank you, sir.
Otherwise, you cannot also show that I have my dad who is a millionaire, spending, no. Then it opens to, not somebody, it is open to something, chaos. So here is institution has to select somehow and then send their money to support you to come to NASA's institution. That's called JVSRP program in JPL, that's called JPL Visiting Scientist Research Program. So we can get them. So talk to your mentor, find the money, and then you get the ticket to the moon.
Thank you so much, sir. I have one more question. So is there any platform so that directly we can connect with the current ongoing studies which is in NASA? Like you have shown some videos in spaceship and that, how you collected the sample from spaceship. So is there any platform so that directly we can watch these videos and we'll get to more information?
Simple answer is no. Because if you are going to request some sample from the PI has to be thought of about such kind of things ahead of time. He cannot have ad hoc situation saying that I have these samples that has to be sent to India for this institution to do it. No, it won't happen. Mainly because of the interagency has to approve things. So it is not at the scientist level, it is in agency level. ISRO will come into thing. It is not ISRO's ego, it is the ISRO's thing, say, oh, I want to do a similar thing, why should I do it with NASA, kind of thing, okay? So there are a lot of things are there that way. The other one is NASA being public money, we have to put all our data into public domain, open to the humankind. Okay, not only US, humankind. Now the situation is different. It is digital data. So digital data out there that a lot of people can work on it and then find intelligently a different way, and then you can publish that. Those are the informatics, bioinformatics, all these things can happen. That's what we have, NASA Gene Lab. So make a note of it and then go into Google, NASA Gene Lab, you will find it. So you can do that kind of informatics work without any issue.
Okay. Thank you so much, sir.
Any more questions?
That's all?
Okay. Thank you, sir, for taking us into the wonderful visualization and imaginary world, displaying a lot of like how the microbial life exists in the space. Also giving a gist of the different microorganisms or the fungal strains which are present and how they grow, or about their biofilm or the exopolysaccharides. Also, we got to know about how the morphology of the microbes varies with microgravity, and the wonderful technology, the Omics technology, how it works in the spacecraft. So I guess you also gave us the future scope, how we can collaborate with you in terms of microbiology or any omics work we can do. I guess most of them over here might be interested to collaborate with you too.
Channelize all those things through Dr. Rekha because she knows where I am.
We would now like to invite Dr. Arun Bhagavat, Principal, Yenepoya Institute of Arts, Science, Commerce and Management, Mangalore, to honor our chief guest with token of love and appreciation.
Thanks a lot.
Thank you, sir. On behalf of Yenepoya Research Center, Yenepoya University, I would like to extend my heartfelt gratitude to our esteemed guest, Dr. Kasturi Venkateswaran, for giving his prestigious time and sharing his research experience and motivating the young minds. I would also like to thank one and all the faculties and dean of principal of the Yenepoya Institute. Thank you one and all present over here for making this a wonderful day and a memorable day. Thank you, sir.
Thank you very much. Hope they enjoyed.
Definitely, they would. Otherwise, they wouldn't have asked that many questions.
No, it's not long. I don't want them to get bored. That's